Hans Lambers is today's speaker. He's, I would say, probably one of the most influential plant ecologists of the decades. So beyond that, he probably needs no introduction. Hans is Professor of Plant Ecology at University of Western Australia. He's also a distinguished professor at China Agricultural University, and he's recently had the honor of becoming a life member of the Australian Society of Plant Scientists. You've seen his name many, many times on publications. That's because he's published over 600 journal publications, a truly astounding number. As well as textbooks. I'm sure we all have a copy of his physiological ecology book somewhere on our shelves, or to these days somewhere as a PDF on the computer. And beyond that, I'm sure you know his name as being editor-in-chief of Plant and Soil. So Hans is going to talk today about why are tall eucalypt forests in southwestern Australia, biodiversity hotspots, not exhibiting high plant biodiversity. So Hans, the floor or the screen becomes yours.
Well, thank you very, very much for your very kind introduction. I hope I can live up to the high expectations that you raised. If not, then I just blame you for all of that. I moved to Australia in 1998, and I was astounded by the tremendous biodiversity that I found there. And one of the things that really astounded me is that the number of non-mycorrhizal plants in that part of the world is really incredibly high. And so I had lots of questions to ask. Today, I want to talk about a new project that we started only a few years ago, and that is, why are the tall eucalypt forests in the southwest biodiversity hotspot, why are they not exhibiting a high plant diversity? If you go to those tall forests, the Kerry forest, then they are majestic. They are really very impressive, but what they are not is very biodiverse. So I was really puzzled by what was the case. But before I can answer that question, I need to tell you a little bit about why our biodiversity hotspot is so diverse. And that's what I'm going to begin with. And then I'll go to my Kerry forests and other forests. So here we see the original 25 biodiversity hotspots in the world. And you see the southwest of Australia is number 12 over here. Right now, we acknowledge that there are about 36 biodiversity hotspots in the world. It doesn't really matter. The southwest of Australia is clearly one of those 25 or 36. And if you want to know where that biodiversity actually is, then I want to talk about the vegetation types. In dark green at the bottom here, over here, where the rainfall is highest, we have the tall forest. They are the Kerry forest and the Tingle forests, and they are among the tallest trees in the world. Absolutely fabulous, really impressive, but not diverse, as I will show in a minute. Then that lighter green, they are the Jarrah forests, still not particularly diverse. And then we go to the woodlands over here, and the yellow, that is what we call the Congan. That's the heath vegetation. Now that is where we have our greatest biodiversity, as shown in this slide over here. The rarer it actually is, the greater the biodiversity, the greater number of species that we find over there. And we have three iconic national parks: Lazur National Park over here, Fitzgerald River National Park, where I will go when I come back from Europe in March, and then Stirling Range National Park. Those are the iconic national parks with an enormous biodiversity. But you can also see that Greater Perth has a tremendous biodiversity, as you can see over here. If you want to know where we find the endemic plants, we can see that the endemism is also greatest in these three iconic national parks. And again, Greater Perth also has a lot of endemic species. So clearly typical, where we have those tall forests over here, those eucalyptus forests, that's not where our biodiversity is. It's not low diversity compared with the beech forest and the pine forest that you may be used to in Europe, but it's very low diversity when it comes to the diversity that we have here in Western Australia. Now, what are actually the factors that determine plant species richness? Well, the geological stability is really very important. If your landscape gets inundated by lava all the time, well, that really destroys the biodiversity, and the evolution has to start all over again. Climatic stability is also very important. If you have glaciers, they work like bulldozers. They destroy all the vegetation. Well, in this part of the world I'm talking about, we haven't had glaciations for the last 250 to 300 million years ago. So well before all the species that are part of our biodiversity actually came to life. And the third aspect, and I really want to emphasize that as a plant scientist, is that phosphorus is extremely important. So the biodiversity that we see here in my bottom slides, the biodiversity here is really incredibly high. If you have a quadrat of 10 by 10, you easily pick up 40, 50 or more species. And you move your quadrat, you pick up another 40 to 50. So this biodiversity is not in those tall forests, but it is in this low heathland over there. And they are incredibly infertile, particularly low in phosphorus over there. But clearly for an ecologist, biodiversity is more than lots of species. It's also about patterns and processes. And it's those patterns and processes that we actually want to talk about today. And one of those clear processes is that if we measure the plant species richness, we can really see it sharply increases with decreasing soil fertility, with decreasing plant phosphorus concentrations. That may be a bit counterintuitive. Well, I'll explain to you how it all actually works. And what is really also very intriguing, that is this red line over here. And that red line represents Proteaceae, one of the families that I have focused on over the last 25 years. And what is special about the Proteaceae is not only that they look very pretty, but what's very special is that is a non-mycorrhizal family over there. So here we go. We go to a part of the world where we have much poorer soils than you are used to in Europe, and the importance of mycorrhizas is far less important than what you are used to. Before I moved to the University of Western Australia, I was a professor at Utrecht University in the Netherlands, and I taught my students that mycorrhizas were very important to get phosphorus for the plant. And all of that is true, of course. I'm not going to deny that. I'm not going to tell you that I told lies to my students in Utrecht. Definitely not. Mycorrhizas are important to get phosphorus. But now we go to a part of the world where soils are much poorer than what you can imagine in Europe, and yet the non-mycorrhizal species are far more important than they are in the rest of the world. There's one important message is, if you want to conserve that biodiversity here in our biodiversity hotspot, or if you want to create it in your own garden, like I'm very keen on, I've got a native garden, then you must keep the soil phosphorus concentration to an absolute minimum. If you eutrophy the environment, you lose a lot of your biodiversity. Now, you all know that mycorrhizas are important to get phosphorus for the plant, and in exchange, they give sugars and fatty acids. So the bread and butter is provided by the plants, and they give phosphorus to those plants. Don't get me wrong, mycorrhizas do occur in Western Australia. I'll give you some examples in a minute. But my point is, this symbiosis is not really as important as it is in the rest of the world. Here I'm quantifying that. If you look at all plants across the globe, we can see that only 18% of all those plants are non-mycorrhizal. And a lot of them have a symbiosis with arbuscular mycorrhizal fungi. Now go to the right. The biodiversity hotspot I'm talking about. With much poorer soils, we see that the non-mycorrhizal fraction of all the plants is now 40%. We've got less arbuscular mycorrhizal plants and a bit more ectomycorrhizal plants, but that is because we've got a woody vegetation, and so the ectos become more important than the arbuscular mycorrhizal plants. So we do have arbuscular mycorrhizal plants, and here are some families and some species that really exhibit these arbuscular mycorrhizal symbioses. We also have ectomycorrhizal symbioses, in particular in Myrtaceae, for example, and they tend to have an association with both arbuscular and ectomycorrhizas. So I'm definitely not telling you that there are no mycorrhizas here. We've got plenty of mycorrhizas, but they are not nearly as important as they are in your part of the world. So why is it that we have so many of these non-mycorrhizal species? I talked about the Proteaceae at the left here already. We've got lots of carnivorous plants at the right over here. And then in the middle, we've got the Hemiodraceae and the Cyperaceae. We have got lots of non-mycorrhizal plants in a large number of families over there. So what features do they actually have? Well, here you see a plant that we grew in nutrient solution, and you see that it makes wonderful photogenic cluster roots over here. If you dig them up from the soil, they look like this, a bit dirty, but you can really see those cluster roots are not just a feature in nutrient solution, they are very common in their natural environment as well. The Banksias look even weirder. They look more like Christmas trees rather than bottle bushes. And if you dig them up from the soil or in a pot, we can see what they look like over here. And they are the secret why these non-mycorrhizal plants are so successful in these very nutrient-poor environments. So if you want to work out where the Metaceae, like the eucalypt, for example, where they grow, well, they prefer the somewhat richer soils. None of these soils are rich compared to European standards, but they prefer the somewhat richer soils. But you can also see there are still plenty that actually grow on these poorer soils over here. What is also very clear is that the Proteaceae, like the Banksias, which I've studied in great detail, and also the Hakeas and Grevilleas, they really prefer the poorer sites. In fact, if you give these plants more phosphorus, you're likely to kill them. So they've got a different distribution pattern. And why is that? Well, for that, to answer that question, we really need to study their physiology. And we study these cluster roots that I showed you before, and they are remarkably ephemeral structures. They only live for about 3 weeks, and then it's curtains. Then they die. If you only live for such a short period, then you've got to grow very fast. And if you grow very fast, it's just like when we run very fast, you've got to respire very fast. And that is exactly what they do in these early stages over here. But then the respiration slows down. What do they then with their carbon? Well, they turn that into carboxylates. And these carboxylates, they release in their environment. And those carboxylates, they are the secret why these plants are so successful, because they are actually the ones that mobilize that tiny amount of phosphorus that we find in these soils. So that's the cartoon that I'd like to use to explain that. So carboxylates are released by a carboxylate channel that has been studied in great detail in white lupins. And in the soil, the carboxylates take the place of phosphorus. Phosphorus is bound to soil particles. That can be organic phosphorus, that can be inorganic phosphorus, but it's all bound, absorbed to soil particles. The carboxylates are negatively charged, just like phosphate, and there's just an ion exchange. The phosphate is pushed into solution, and that is then taken up by these plants. So you can really see these cluster roots, they wrap themselves around soil particles, they wrap themselves around these gravelly stones over here or around limestone, and they even get phosphorus. We showed that. They even get phosphorus out of these stones over here. Mycorrhizal plants can't do that. They're great scavengers. They get phosphorus that's far away from the root surface, but they can't get phosphorus that is tightly bound to soil particles. But these cluster roots, they can. And so, what is the secret of these non-mycorrhizal plants? They have specialized roots, and they release vast amounts of citrates and malate. And citrate and malate mobilize the phosphorus that is in the soil. That's actually a mining strategy. So it's not a scavenging strategy, as we know for mycorrhizal plants. It is a typical mining strategy. And now we can understand why the scavengers prefer the soils that are somewhat richer, where there's more phosphorus in the solution, whereas because they have their scavenging mycorrhizas. But the miners, they need that mining strategy. They really need to mobilize the phosphorus, push it in solution before they can take it up. So totally different strategies. And I read this morning in a technical review that this is a weird strategy that you only find in some soils in Western Australia, and it's not relevant anywhere else. That was the implication. they could not be more wrong. First of all, it's not just a handful of species that occur in nutrient-poor soils. We've got hundreds of these proteaceae, and they all grow in this part of the world, in this biodiversity hotspot. To actually say this is an anomaly, this is an exception, that is actually blatantly wrong. On top of that, it's not just proteaceae doing that, it's also cyperaceae. They have their dorsiform roots. They do exactly the same. And those dorsiform roots, they are most certainly not restricted to the southwest of Australia. They grow in your own backyard. And they tend to be growing in those areas in Europe, for example, where the phosphorus availability is low. There may be enough phosphorus, but the availability is low, either because the pH is high, your chalk grasslands, for example, you may have enough phosphorus, but it's very poorly available. But these cyperaceae with their dorsiform roots, they release carboxylates, they can get hold of that phosphorus. Or you go to the acidic grasslands. Again, lots of cyperaceae over there. And again, that's where the carboxylates also work. So it's wrong to think that this is typical for that weird part of Australia where I happen to now live and where I have studied in great detail. But the same strategy you also find in Europe. We've done a lot of work in China. We've done a lot of work in Brazil, for example. So don't think that this is an odd strategy that you will find nowhere else. That's a wrong thing to think. But you can also see that there are species among the Myrtaceae that actually grow where only the mining strategy is supposed to work. And yet some Myrtaceae do grow there. So the question is, how can those Myrtaceae actually grow there? Well, we studied that in somewhat greater detail because we are really very keen how this Scholzia, which is a mycorrhizal species, and mycorrhizas are not very good to get phosphorus out of these very nutrient-poor soils. We were very keen to work out how this Scholzia species, this Myrtaceae, could actually function there. And we decided to grow them together in a pot and study facilitation. So we were hypothesizing that the Banksias with their cluster roots would mobilize phosphorus and other nutrients, and that the Myrtaceae, the Scholzia involucrata, would actually benefit from that. And indeed, that is exactly what we did. So here we've got the phosphorus mobilized by these cluster roots, and they do that to help themselves, of course, but the neighbors, they are very grateful to also use some of that phosphorus mobilized by these plants. So in a nutrient-poor environment like this, facilitation is the name of the game. It is far more important than competition. I'm not saying that competition plays no role. What I am saying is we've got to look at facilitation when we look at such a biodiverse system. And that helps us understand why we can have these Myrtaceae co-occurring with the Proteaceae, although they do not have a superior phosphorus acquisition strategy. But nobody can be good at everything. There's always a price to pay for being good at something. And the price these pay, we thought, might be that it might not be very well defended against pathogens. And the pathogens I want to focus on is actually this oomycete over there. And they are not introduced oomycetes. They are actually the native oomycetes that you can pick up in our native system over here. And this picture actually shows it up. It summarizes it quite neatly. And let me just focus on these two photos over here, because one picture is worth 1,000 words. Here you see one of our Banksias, so Proteaceae, with their cluster roots, grown in its own soil. And being a plant scientist, I don't like pathogens, so we always pasteurize our soil. But then I started collaborating with an ecologist, and they, for some odd reason, they seemed to like pathogens. So they were keen not to pasteurize the soil. And I'm glad I started my collaboration with them, because we really discovered that if we do not pasteurize the soil, this poor Banksia does not grow very well at all. And that then led us to the secret why we have such a high biodiversity. Because my cartoon in the previous slide was not quite correct. It was incomplete. That cartoon pretended that there were no pathogens. And I showed you already what happens when you have no pathogens. The Proteaceae have a superior phosphorus acquisition strategy, and the neighbors actually benefit from that. But the reality is that we do have pathogens. And those pathogens, they really affect the proteaceae. I showed it in my previous slide. They really affect the proteaceae to a major extent. But when it comes to mycorrhizal species, the role of mycorrhizas in this system is not, as I pointed out already, is not to help the plants getting phosphorus. It is actually very important to defend the plants against pathogens. So these plants do well because they are mycorrhizal. Those mycorrhizas are pretty useless when it comes to getting phosphorus, but they help the plant to get the phosphorus. And now you can see why we have that tremendous biodiversity. It is based on plants with this superior phosphorus acquisition strategy, but they're not very well defended against pathogens, and they co-occur with plants that don't have a particularly impressive phosphorus acquisition strategy, but they are much better defended against pathogens, courtesy of their mycorrhizal fungi. So these friendly mycorrhizal fungi, they protect their mycorrhizal host against the pesky oomycete pathogens. And together, these four players, they account for that tremendous biodiversity over there. So let me summarize that part of the story and then get on with my eucalypts. We've got the greatest biodiversity where the soils are poorest. And that is where you need that superior phosphorus acquisition strategy. That's where you need the cluster roots. And facilitation among plants in that nutrient-poor habitat is far more important than competition. Competition does occur, absolutely, but facilitation is really the name of the game. And the proteaceae are very efficient at getting the phosphorus, but they are not very good at defending themselves against the pathogens because that's where they need the support of the mycorrhizas. The mycorrhizal plants are not very good at getting the phosphorus, but they cope better with pathogens courtesy of their mycorrhizal partners. And the interactions among the four players accounts for our mega diversity in the Southwest Biodiversity hotspot. But then that leads to my final question that I want to address now is, why are some of our impressive, majestic, tall forests where we have the cherry trees, why are they not as diverse as the sandplain flora that I'm capturing here in this slide over here? Well, for a while, I thought, and lots of people actually thought, these cherry forests, these forests that I'm showing you over here, they are probably a little bit richer. And I started off by saying that if the soils are richer, the biodiversity is less. So is that really the case? Are the cherry forests and the yarry forest pictured in these two photos over here, do they really have soils that are richer? That was the question. Now, of course, there's only one way to answer that. That is to collect soil samples and measure the amount of phosphorus in there. Now, I wasn't the only one thinking that these soils were richer. We had a politician 100 years ago, Sir James Mitchell, but the press referred to him as Mukau Mitchell because he sent beautiful postcards to England and he told those people, come to Western Australia because we've got these rich soils and look at them. You can have your dairy farm over here. Get rid of those trees. They are just useless, but you can replace those trees by a dairy farm and then you'll be rich and famous. But I can tell you, it was a complete failure because they soon discovered soil nutrient deficiencies and they were a killer to the crops and the animals and all the farmers went broke. So Mukau Mitchell had it absolutely wrong. So what would happen if these mycorrhizal plants, this beautiful cherry tree, for example, they grow up to 100 meters tall. What happens if these mycorrhizal plants would discover during the course of evolution the trick to release carboxylates and mobilize phosphorus? Now that would make them very strong competitors, of course, because now they can have access to phosphorus just like the Proteaceae and the Cyperaceae and the Hemoderaceae, but they are still mycorrhizal, so fungi would protect them against nasty pathogens. These cluster roots, they release carboxylates, and these carboxylates, they are released to mobilize the phosphorus, but in that process, they also mobilize manganese, and that's cool, because the uptake of manganese in plants is not tightly controlled at all. So whenever plants mobilize manganese in the rhizosphere, you will end up with plants that have high leaf manganese concentration. So I started using that as a proxy to work out what plants do below ground. Measuring carboxylates in the rhizosphere in the fields, in the forests, is a nightmare. You can waste a lot of time and get nowhere. But if you can use this proxy, then you are in business. So what did we do? We went to this Yari forest after we discovered that the soils were just as poor as those sand plain soils, and we wanted to know whether they did release carboxylates. So what we did is we used two reference species, referent plants. One of them has beautiful mycorrhizas, that's Banksia grandis, and the other one never releases carboxylates, that's this grass tree, Xanthorrhea pricei over there. But we are interested in that target species, in this case, Eucalyptus patens, and we wanted to know whether it might release carboxylates. Well, so we started off measuring the leaf manganese concentration, and you can see it's not quite as high as in the Banksia, but it is a lot higher than in my negative reference. So this suggests, but no more than suggests, this suggests that it was likely releasing carboxylates. So we then started growing them in nutrient solution. We collected the exudates, and lo and behold, this plant did release carboxylates. Not citrate and malate, those were not the major carboxylates like in Banksia, but it was mainly oxalate. But all these carboxylates, they basically do exactly the same in soil. They all mobilize carboxylates. They all mobilize via ion exchange. They all mobilize phosphate. So clearly, this eucalyptus, although it was mycorrhizal, it actually depends on carboxylate release to get the phosphorus. And it has to, because these soils are just too poor for mycorrhizal fungi to be very effective. And then we thought, well, let's look at the other species in the same environments. And we measured the exudation rate of all these plants that had reasonably high leaf manganese concentration, and we found that they all released carboxylates. And in fact, Kerry, that does not grow in that habitat, but a bit further south, also in a high rainfall area, Kerry, when you grow that in nutrient solution, also releases carboxylates. So all these plants that everybody tells me depend on mycorrhizas to get phosphorus, what they really do is they release carboxylates to get the phosphorus. I'm not saying the mycorrhizas are not important. I think they are very important. They are just not important to get the phosphorus, but they are likely very important to defend the plants against pathogens or other forms of stress. So now we can understand why we don't have a very high biodiversity in Kerry and Yari forests. These species, they have traits like the carboxylate-releasing phosphorus-mobilizing proteaceae. So they know how to get their phosphorus. They may not be quite as good at it as the proteaceae, but they are pretty good at it, and so they grow like a bat out of hell in these environments. But being mycorrhizal, they're also defended against plant pathogens. That's where the mycorrhizas come in, not to get phosphorus, but to defend themselves against pathogens. These guys don't need facilitation. They don't need to depend on the proteaceae to mobilize the phosphorus. So now we can expect these species to be very strong competitors and basically compete with all the plants that cannot release carboxylates in that environment. They have the best of both worlds. They know how to get their phosphorus, but they also know how to be defended against pathogens. So you can expect them to be very strong competitors, and then the biodiversity actually plummets. And clearly, Mukau Mitchell was totally wrong. His scheme was a complete failure, and what we learned from that is there is actually a role for science underpinning political decisions. Well, of course, this was 100 years ago. Times have changed. Now, if scientists say we must be careful with releasing more CO2 because it affects the climate, then immediately politicians rush up to ecologists and other people, and they ask, what shall we do and how fast should we act? If only that were true. So I think we still have a long way to go before science really has the effect that it should have on the wider community. Now, we can now ask our questions, are these carboxylate-releasing eucalypts, the two I was talking about a minute ago, are they Darwinian demons? And with that, we mean Can they do everything very well? Are they both good at getting the phosphorus and good at defending themselves? They're likely very strong competitors, and they would also compete against the Fabaceae that do not release carboxylate. Some do, but most of them in that environment do not. So if they compete very strongly with the Fabaceae, they basically deprive themselves of a source of nitrogen, because in that environment we are looking at, we lose nitrogen all the time. It's rather wet, so we lose it through denitrification, we lose it through leaching, and we also lose it in my part of the world through fire. Ash returns to the soil, but nitrogen does not enter up in the ash, it all goes up in the air. So we lose that nitrogen all the time, so these nitrogen fixers, including the Fabaceae, are crucially important to keep the nitrogen in the system. Now, that basically means that these eucalypts I was talking about, they function at a very low leaf nitrogen concentration. And that basically means that they have a pretty low photostatic capacity. And what these plants do is to compensate for that low photostatic capacity, they open their stomates further so they can use as much as possible of that low photostatic capacity. But that only works if there's plenty of water. You can't do something like that in the desert. And so there is a trade-off between water use efficiency and nitrogen use efficiency. You either are very efficient at using your nitrogen and phosphorus, or you're very good at using your water very efficiently. And that basically, this trade-off basically prevents them from becoming Darwinian demons. And I show you a very nice slide, which I took a few weeks ago when I asked a friend of mine, why are all these yarry trees, Eucalyptus patens, why have they all died? And he explained very clearly what had happened. In the 1980s, a neighbor decided that he wanted water for his irrigation of his orchard. So he basically blocked the water flow that led to this environment, and all the yarry trees in this environment that were unable to control their stomates, they simply wanted to get on with transpiring a lot, they basically all died. Other species, including all the eucalypts in that environment, did not die. So it's not so that all species in that environment have a poor stomatal control, but this yarry tree that is releasing massive amounts of carboxylates and loses a lot of water to compensate for its low nitrogen concentration, low photostatic capacity, that species has very poor stomatal control, and all these plants actually died. And for another eucalyptus species, Eucalyptus wandou, colleagues of mine did a very nice project where they showed that after a few dry winters, this Eucalyptus wandou was in dire straits, and it did not die, but we saw tree crown decline. But then after the winter started becoming wetter again, then they basically recovered. But the story is basically the same, that these species that release a lot of carboxylates, they pay a price for that, and they lose a lot of water. Then, of course, the question came, do all eucalypts function like yarry and curry? And if you have a quick look at this table, the answer is very similar, definitely not. There are some that have very high leaf manganese concentration, and all of those that we tested in the glasshouse, they all release carboxylates. We also have one that have very low leaf carboxylate concentrations, and all of those that we tested in the glasshouse, they release very little carboxylates. And then there's some where it is actually dependent on the site, and there are also some where it is actually rather variable. So I want to explore with you why some eucalypts fit in one category and other eucalypts in another. And so what we do is we go to the field. We have a wonderful excuse to go to one of these national parks and collect some leaves over there, and we measure the leaf manganese concentration. And we do that by having a positive reference. We quite often use a Banksia for that. And we use a negative reference. We prefer the grass tree over there, and these are our test species. And we do that for a whole range of different sites over here, and we have now collected data for about 40 species over there. So the story is coming together. We also measured leaves, the leaf manganese concentration of leaves from our UWA herbarium, And there is a bit of a story that is a bit of a warning for those who may be interested in using herbarium material, because there is a little bit of a trick. The ones that have high leaf manganese concentrations, like Eucalyptus decipiens, for example, over here, that also has high leaf manganese concentration in the field. So that is fine. And there is, of course, Eucalyptus patens. We mentioned that already. Our herbarium material also had a high leaf manganese concentration. But then we've got species like Eucalyptus wandoo in the field, always at different sites, always a high leaf manganese concentration, and in herbarium material we can't actually detect it. And the same is Eucalyptus loxofleba, always a high manganese concentration, no matter which site you go to, but in herbarium material, manganese is not detectable. Now, we thought about that, and here is a bit of a warning if ever you want to use herbarium material. In the herbarium, the taxonomists, they tend to collect specimens, and they have nice young leaves, and usually very few old leaves. And if you can see in this manganese hyperaccumulating Grevillea species, this is not from Australia, this comes from the literature, you can really see that young leaves have much lower concentrations. That's also true for this hyperaccumulating species. When you go to our own non-hyperaccumulating eucalyptus species, we can also see an enormous difference between the old leaves and the young leaves that are still expanding. And in fact, sometimes when we can't find that Xanthorrhoea pricei that we'd like to use as a negative reference, we actually rely on picking young leaves, and we use those as a negative reference, and that actually works fine. So the problem with herbarium material is, if you find high values, you're fine. But if you find low values, well, you think you may still have to go to the field to actually collect them there, because you may not have had the right leaves in those herbarium specimens. There may not have been the leaves that you really want to measure leaf manganese, because the young leaves don't accumulate it. It's only the older leaves that accumulate it over time. The different groups of plants, as I pointed out, and I only give you one example, but I have lots of others, but there's no point showing you all these beautiful pictures. Some of them always have a high leaf manganese concentration, no matter where you sample them. But these, as you can see over here, these eucalyptus stands, they tend to have a very low diversity understory. And all these plants that we then measured in nutrient solution, all these mycorrhizal species, they depend on carboxylate release. The high leaf manganese concentration gives us a clue, but we follow that up and really measure that in nutrient solution. So all these plants with a low diversity understory, you can be quite sure that they all depend on the release of carboxylates and not on mycorrhizas. Now, there is one thing that is rather peculiar about these eucalypts, because you can give them more phosphorus, and that does not change their release of carboxylates. If I would show you data for lupins, white lupins, for example, or for one of our Proteaceae, you would really see that as soon as you give them 20 micromole of phosphorus, let alone 40 or 80 micromole of phosphorus, they would immediately stop releasing any carboxylate. They are smart. They stop releasing carboxylates. They even stop making these photogenic cluster roots. Eucalyptus does not do that. So in eucalyptus, the release of carboxylates, unlike that in Proteaceae and white lupin, is not controlled by the phosphorus status of the plants. Those high leaf manganese eucalypts, they possibly release carboxylates not in response to low phosphorus status, but they probably do that to remove a surplus carbon. That is the current thinking. Whatever controls carboxylates, though, the carboxylates, they will mobilize phosphorus in the environment, of course, and they will also detoxify aluminium. Whether it's controlled by aluminium or controlled by phosphorus is neither here nor there. In the soil, they will mobilize phosphorus and they will detoxify aluminium. It's just not controlled by aluminium or by phosphorus like it is in a range of other species. And these eucalypts, they likely have very poor stomatal control, and that, we think, triggers a surplus of carbon. So they tend to have a low water use efficiency, and that Eucalyptus patens basically dies when you deprive it of its natural water supply. So when we look at the whole range of eucalypt, you can see that there is a difference. The high leaf manganese concentration, the plants that have leaves with high leaf manganese concentration, they definitely have a lower water use efficiency than the ones that have a low manganese concentration. So there is clearly a highly significant difference, but I hasten to add, the variation is also enormous. So there is still something that we don't quite get. There must be more that I don't quite understand yet. They do have a low water use efficiency, but the variation is such that I think that release of carboxylate is probably controlled not just by poor stomatal control, but possibly by something else as well. But all of that is work in progress. Then we go to another eucalypt, Corymbia ficifolia. And this one has a variable leaf manganese concentration. So you go to one side and you collect a number of species, and one of them is high, one of them is a lot lower than that. It's all relatively high, higher than the reference species, but it's not like in that previous group where it was definitely all high. Here it changes, it varies a lot more at one particular site. And all these eucalypts, they have a very biodiverse understory. And they likely depend on facilitation by carboxylate releasing neighbors to get that P. But it all depends on how close they are to the neighbor. And we haven't actually studied that for this particular species, but we know that for this acacia species over there. This acacia species does very well after a fire, and then it germinates massively, and a lot of species, they die after a few years, but some hang in. And then we measure the leaf manganese concentration in this acacia, and we measure it as a function of distance to this big banksia that makes these weird and wonderful cluster roots over there, mobilizing the phosphorus. And we can really see the manganese concentration in this species that does not release carboxylate itself is high when it's close to one of these carboxylate-releasing species, and when it's 10, 15 meters away, you can really see that the manganese concentration is much lower, as low as of the negative reference over there. So we think the variation that we find for eucalypts is basically due to the distance to the nearest facilitating species over there. And then the other group is they have a leaf manganese concentrations that depends on location. So sometimes it's high and sometimes it's as low as a negative reference. And here we think that facilitation may actually involve mycorrhizal hyphae because they actually intercept the manganese. So mycorrhizal species never have high leaf manganese concentration, but sometimes it also may be facilitated directly, not involving those mycorrhizal hyphae that would intercept the manganese. And we actually tested that in the laboratory, and we are doing further work to actually test that in greater detail. Here we've got a monoculture of this Proteaceae with cluster roots. Here we've got a monoculture of the eucalyptus, the mycorrhizal plant. And here we grow them together. And these were grown with or without ectomycorrhizas, but of course this is a non-mycorrhizal plant, so those mycorrhizas did nothing. And here we grow the eucalyptus with and without mycorrhizal fungi. And in this case, they did establish a mycorrhizal symbiosis. What were the results? Well, if you grow these plants alone, these banksias grow very well because they can cope with very nutrient-poor soils. No mycorrhizas are required for them at all. If you grow the eucalyptus in that very poor soil, it grows very, very poorly because, as I said before, these soils are just too poor for those mycorrhizas to be very effective. But if you grow it in one pot together with a banksia, well, their biomass actually shoots up by 51% at the expense of the banksia. So they compete with the banksia, and the banksia actually produces 30% less. And if you repeat that experiment, but now with the ectomycorrhizal fungus, there is less of an effect on the banksia, but there's still a very positive effect on the mycorrhizal plant, the eucalyptus. So this plant can really be facilitated without but also with mycorrhizas. And that, we believe, explains the variation between sites. If sites are very poor, mycorrhizas don't get a chance, and there is no way that these eucalypts can actually be facilitated involving mycorrhizas, and then you end up with a high leaf manganese concentration. If the soils are slightly better, then mycorrhizas do have a chance, and they step in and they pick up the phosphorus mobilized by the neighbor. And in that case, they intercept the manganese, and you end up with a low leaf manganese concentration. So when you have all this variation among sites, not within the site, but among sites, it is probably, they do depend on facilitation, but it may or it may not involve mycorrhizal hyphae over there. And then we've got species where the manganese concentration really varies depending on site. In this particular case, it would be facilitated by the neighbors with mycorrhizas, but at other locations, it could be facilitated without involving mycorrhizas. My conclusions then, mycorrhizal plants in megadiverse sites, they depend on facilitation by their non-mycorrhizal carboxylate-releasing neighbors. And those 4 players I was mentioning, they explain why we have this megadiversity in the Southwest biodiversity hotspot. Mycorrhizal carboxylate-releasing plants, they have a low diversity understory. I showed you that for some of the eucalypts. And they are likely very strong competitors against plants that do not release carboxylates and against non-mycorrhizal plants, but they are not Darwinian demons. The carboxylate-releasing phosphorus mining strategy is more effective than a mycorrhizal scavenging strategy when soils are extremely poor, or when the soils are not poor, but the availability of phosphorus is very low. And that could be the case in strongly P-sorbing soils in the tropics, for example. They may not be super poor, not as poor as the soils I was talking about, but the availability of phosphorus is definitely very low. And it could also play a role in your chalk grasslands or your acidic pastures, for example, in Europe. Most vestibular plants can establish a mycorrhizal association, but the very fact that plants belong to a mycorrhizal family, the very fact that you can measure that they have been colonized by mycorrhizal fungi is no evidence that their phosphorus acquisition depends on those mycorrhizas. Those mycorrhizas may play a very different role, and they may do nothing in terms of phosphorus acquisition. And if you thought I did all of that work myself, then I hate to disappoint you, because a cast of thousands, lots of students, lots of colleagues, lots of international collaborators, they played a very important role. And if it wasn't for Don and John Williams, and I have misspelled their name, I will correct that. If it wasn't for them, then we could not have collected so many eucalypts on their farm. So we are also very grateful for their input. And of course, the Australian Research Council. Thank you.
One of the things I found really interesting is slightly aside, but do you get a build-up of organic matter in these sites? Do the sites differ in organic matter? When you have this massive turnover,
it depends on where you are. When I go to our dry sites, the productivity there is so low, there is no organic matter. And the microbial breakdown may not be very fast. But if you go to Mediterranean environments with high light intensity, then the breakdown by light tends to be far more important than the microbial breakdown. So there is basically no litter layer there. When you go to the south, where the rainfall, so the site I was just talking about, the rainfall is about 550 millimeters. When we go to the south, the rainfall is 1200 millimeters. Then there is a buildup of litter. Yeah, so there is quite an important difference there. And because of the buildup of litter, you also promote the oomycetes. And it's very interesting. When you go to those high rainfall sites, we find very few proteaceae. So when we go to our northern sites, we find at our sites 100 or more proteaceae. When I go to my southern sites, 1200 millimeters rainfall, I only have about a dozen. And I think the oomycetes will have wiped out those proteaceae. But what is very interesting with those proteaceae that do occur there, they are all associated with mycorrhizal fungi. You always find mycorrhizal structures in those roots. And you never find that when you go to the dry sites. And it basically shows if you want to grow in those wet sites, then you'd better get help from mycorrhizal partners. And if you don't get that help, then you get wiped out.
Yeah, I just follow up my own question. That's exactly what I was wondering. I was wondering if on these sites where you get more organic matter, the mycorrhizas play a greater role in organic matter accumulation than weathering processes, as it were. And certainly you'd expect that you get more pathogens with more soil organic matter. So yeah, the question is, do you think those few protozoa growing in less diverse places learned the trick the other way, forming mycorrhizal associations?
Well, that was basically what I was trying to tell you when I was talking about our wet side compared to our dry side. And the answer is yes. In those less diverse sites, we look at a range of chronosequences, and one of them has a 1200 millimeters rainfall pattern. And as I said, when we go to the dry chronosequence, we find 100 proteaceae. When we go to the wet chronosequence, there's only about a dozen or so. But of all the species, the proteaceae species that we looked at so far, we haven't studied all of them yet, but the ones that we have studied, they all have become mycorrhizal. And those mycorrhizas would do nothing for their phosphorus uptake, but they would, I think, be very important. And we're testing that now in glasshouse studies. One of the PhD students is doing a wonderful project on that. We're testing now in the glasshouses if indeed they are protected against those oomycete pathogens. So yes, they have definitely learned that trick. And we are now following that up in glasshouse study and trying to work out how good are they at playing that trick.
If you're using manganese as a proxy, this is from Boyan Singh. If you use manganese as a proxy, if you introduce new plants, do you have to test them? I.e., could this be universal? And
I think that's actually a very good question. And we have now done work in different continents. We started work in Brazil, and Brazil is very interesting because they have soils that are just as poor, and we see that carboxylates play a very important role. But what is really fascinating for an ecologist is that all these species in Brazil that play the same tricks, they belong to totally different families. And that's fantastic. It basically shows that if you want to grow in those nutrient-poor soils, there's only one trick that works, and that's not being mycorrhizal, that is releasing carboxylates. And different species and different families have all discovered that. So that was Brazil. Then we started work in the Mongolia, in the grasslands there. And again, we use leaf manganese, and we show that these plants, they also release carboxylates. So the manganese proxy also works there. Then we started doing work on the Tibetan plateau, where they looked at global change and they warmed the system. And when you warm the system, your demand for phosphorus increases. If the demand for phosphorus increases, then all these plants that can actually mobilize phosphorus are favored. And you see that indeed the cyperaceae that play this trick, they are actually favored. And you can actually pick that up by looking at leaf manganese concentration. We are just working on the revision of a paper that was provisionally accepted by Journal of Ecology. That's on the grasslands in Uruguay. Again, leaf manganese works there. And I can't see why it may not work in Antarctica, I don't know, but it works on all the sites, different sites in China, different sites in South America. It all works there. And in fact, we are actually scanning a whole range of species in China, and we see an enormous variation in leaf manganese concentration at natural sites over there. And then I think another part of the question was, what if you introduce new species too? And that's also an interesting question, because we have looked at invasive species in Southwestern Australia. And you wonder how invasive species can ever make it in these nutrient-poor environments. And the answer is very simple. They play the same trick as the plants in our native system. So we've got a Britza species, for example. And Britza tends to occur in chalk grasslands. And it does very well here. And it does very well because it plays the same trick as the plants in this natural environment. And it would make perfect sense for that species to do that in its chalk grassland, because to get phosphorus out of these chalky soils, you also need carboxylate. So I think that that principle probably plays a role just as much in Europe as it does in China, in South America, and in Australia.
Is there seasonal variability in mycorrhization and in the mycorrhizal pathogen interactions?
There may well be, but we are looking into that, because we live in a Mediterranean climate with hot, dry summers, and right now it's about 40 degrees here this week, so it is hot. And then we have got wet and cool winters, and you would expect the pathogens to be more important in winter than in summer. We are actually following that up, and we are using molecular techniques to actually work out the pathogen pressure, and we are also working out the pressure in the dry sites compared to the wet sites. So we are working on that. It's a very good question that Boris raises there, but I haven't got the answers yet. But I hope to be there in a year from now.
There's a general trend in a decrease in phosphorus in forest trees throughout most of Europe, and whether this is related to mycorrhizas.
And that is because of your nitrogen. I mean, historically, Europe, being a young landscape, would be nitrogen limited. But of course, with your enormous nitrogen input via acid rain, burning fossil fuel, your input of nitrogen is enormous. And so you enhance the nitrogen supply, but that basically means that you make phosphorus more limiting. So all these tricks that I'm talking about, they become increasingly important also in your forest in Europe. And we are seeing that in China, for example. The N to P ratio in the forest really changes dramatically. N goes up, but P tends to go down. And you basically push these plants to play the tricks that I was talking about. And I bet my bottom dollar that a lot of species in your system will actually switch to that strategy. So to actually think this is something weird from the southwest of Australia, you're making a big mistake dismissing that as something that only the Aussies do. No, that's more widespread. And we are discovering that in China right now. So you're getting behind if you don't jump on the bandwagon and actually study phosphorus nutrition of these forest trees and actually work out what their tricks are. Because you may push them to a system where the mycorrhizas are not effective enough, and that you may actually need carboxylates. And of course, some of your forest species, like ulmus, for example, ulmus is one of these actinorhizal species, and actinorhizal species, as far as I know, they all make cluster roots and they all release carboxylates. So there is one of your, it may not be one of the most important forest species, but it's an example that your species may be mycorrhizal, but I think you've got to keep an open mind and actually realize that they may actually depend on other strategy to get their phosphorus.
I come from Spain, where eucalyptus was imported in the 19th century from Australia. It's adapted really well, especially in northern Spain. The thing that is very controversial is that it uses huge amounts of water and is highly flammable. What would you recommend as a more diverse and less balanced eucalyptus forest in Europe?
I think you've got to be incredibly careful introducing eucalypts to the rest of the world, because they are very thirsty plants. And I've seen that in Brazil, for example, where they affect the water table. They affect the stream flow, for example. You put them in, I mean, the stomatal control of these eucalypts, and I talked about that, is not particularly strong. So whenever there is water, they will use it until they can't use it anymore, and then they may close their stomates. But they are very thirsty plants, so they have a major effect on the water balance of the whole environment. So I would be very careful with those eucalypts, because and there may be, I'm not even sure there are eucalypts that are not thirsty. Most of them are thirsty, but there may be ones that are somewhat less thirsty, but then you would have to look for them.
Is there a difference in manganese uptake between arbuscular mycorrhizal and ectomycorrhizal plants?
All mycorrhizal plants, whether they are ericoids or arbuscular mycorrhizal or ectomycorrhizal, all these mycorrhizal hyphae, they tend to intercept the manganese. And people working on mycorrhizas, they quite often use the low leaf manganese concentration as evidence that their mycorrhizal inoculation actually worked. So I do the other one. I do it exactly the other way around. But mycorrhizal people for years they haven't widely published this, but you hear that from a lot of people that quite often they use that low leaf manganese concentration as evidence that the mycorrhizal fungi properly colonized the plants. So, but they all do that, whether it's ericoids or ectos or arbuscular, they all intercept the manganese. So it works as a wonderful tool to actually work out what they do below ground. But if those ectomycorrhizal fungi don't actually get out, but they stay inside the roots, and the roots actually release carboxylates, then you do have a high leaf manganese concentration. So being ectomycorrhizal doesn't mean that they have low manganese concentration. Mm-hmm. That only is the case if those mycorrhizas are actually actively involved in getting the phosphorus. If they are not outside, if they are just sitting inside because it's nice and safe, and they don't waste their resources going out in the big bad world where they can't do anything anyway, then you will have high leaf manganese concentration, like in my eucalypts.
Thank you very much for a fantastic talk. The series we've reached at least 3 continents this session. We are making the objective of being global. So once again, I thank you very much indeed. That was fantastic.